I'm a very curious person, so I went right online. I went to the Mayo Clinic website where they did have amyloidosis listed, and I was just terribly surprised to hear that it could be a fatal disease. But I think probably my biggest surprise was actually when I went to see Professor Hawkins, and he talked about treating it sick with the fat of chemotherapy, because I felt it's not cancer. Why are you all here?
Welcome all to this Defeat HIV educational webinar. We are so excited to have you with us and look forward to the discussion today and also hearing your comments as well as the presentation today by Dr. Philip Gregory. We're excited to have him join us here on this webinar. Today's conversation we've called Hope, Hype, and Reality. Sangamo talks about zinc fingers in gene therapy studies to control our HIV. It would be a chance. This would be a chance for us all to talk with the people whose research into a cure for HIV has reached. We're really happy to say has reached phase 2 in the clinical trial process. This is extremely exciting. Today's webinar is brought to you by Defeat HIV, our Defeat HIV Community Advisory Board, and we want you to know that you don't have to wear a lab coat to help us find a cure for HIV at all. So a little bit more about the Defeat HIV CAB. This CAB is defined for itself its mission to serve as a communication link and mobilize HIV cure researchers, their institutions, and other communities to work together to cure HIV. And our CAB meets the first Tuesday of each month from 5:30 to 7:30 at the Cal Anderson House, which is located at 400 Broadway in Seattle, Washington. You all are invited to come check us out, those of you who are local, or even if you're not, we have an opportunity for you to join as well, and I will mention that here briefly. We serve dinner each month and offer bus tickets for those who might need assistance with transportation to and from our meetings. You can also check us out, as I mentioned, from your home. And if you have a computer and phone, each month we send out web links for you to use to follow along and to participate in our discussions about the research of Defeat HIV and our mission. So how do we actually do this? By doing a lot of work. We seek to engage people wherever they might be in their understanding of cure research and the difficult obstacles we will need to overcome. We offer webinars such as the one we have joined today. Once again, thank you for joining. We hold special events with researchers to promote access to them, design posters, create videos, glossaries, documents, whatever we think might work to increase engagement of communities in the work to cure HIV. Looking at this collage of just a smattering of some of our community engagement, you get an idea of just how much we've done to date. Now, obviously, as you can see, There we have Francoise, who discovered the HIV virus, and Timothy Ray Brown, we've had the privilege to work with him in times past, and some of our other events there. Our CAB is always eager to welcome visitors and new members to our meetings, and so please consider yourself invited. If you're curious and would like to be added to our mailing list to receive further information about future meetings and upcoming Defeat HIV studies, or simply have more questions about our group, please contact our coordinator, our CAB coordinator, Michael Luella, via his email address, which is mluella@fredhutch.org. That's MLOUELLA@fredhutch.org. I am your moderator. My name is Terrace Mullen. I'm happy to be with you today. I am a Defeat HIV CAB member, as well as a Martin Delaney Collaboratory CAB rep for the Defeat HIV CAB, and I will talk briefly about the Martin Delaney CAB as well. So a few housekeeping things. We can never get around the housekeeping component of these types of meetings. I wanted to review some of the simply important housekeeping rules for this webinar. Number one, we will mute your lines as you sign on. So please be aware of this before you ask your question. Your line is muted. When you do speak, please offer your first name and or city that you live in before asking your question, and this will allow us to better answer your question. Number two, we have two ways that you may ask your question. Number one, you can type your question into the chat feature, which is in the upper right column of the WebEx screen before you. Number two, you can also use the raise your hand feature, which can be found in the lower right-hand portion of your screen. And please know that today's talk will be recorded and made into video, and we will post this in two places. One will be our website, which is www.defeathiv.org, and the other place we will post this video will be on the Defeat HIV YouTube channel. While we're at it, let us briefly let you know about some of the other social media platforms in which you can find us. Twitter. We can be found on Twitter at defeatHIV, and we use the hashtags, hashtag defeatHIV, hashtag HIVcure, or hashtag HIV. Facebook. We have a Facebook presence as well, located at Defeat HIV Seattle, if you're looking for us on Facebook. Please do not hesitate to, quote-unquote, like our posts and our page. It would be greatly appreciated. Thank you. And also, we'd like to keep in mind today that we will only present information that is already in the public domain. And we may defer questions about subjects that Sangamo has not publicly disclosed. So today's focus will be on exclusively HIV. However, we do acknowledge that our industry partner is working on products to treat and cure other diseases. But once again, today our focus will be on their work as pertains to HIV. So a little brief explanation about the Martin Delaney Collaboratory, as I mentioned previously. There are three collaboratories: the DARE Collaboratory, the CARE Collaboratory, and then also the Defeat HIV Collaboratory. These collaboratories have received a sum of $70 million over 5 years to fund public-private collaborations to purge the reservoirs of HIV. A key component of this initiative is the formation of partnerships between academia and the private sector to facilitate the movement of new basic science findings toward eradication strategies that can be evaluated in animals or humans. For the purpose of this FOA, the term private sector comprises large and small, domestic and foreign, profit pharmaceutical, biotechnology, bioengineering, and chemical companies. A little bit about Martin Delaney. He was an activist, a leader, and certainly a public health hero. There are few people who have had a greater impact on HIV/AIDS health policies than Martin Delaney. From the time HIV was recognized as a public health threat in the early 1980s and leading up to his death in 2009, Delaney worked tirelessly as an educator and advocate for HIV/AIDS patients. Delaney was internationally recognized for his work to change domestic health policy by expediting FDA review and approval of HIV/AIDS therapies, successfully lobbying for a fast-track approval process. He played a key role in significantly reducing the time it takes for new and emerging drugs to reach the hands of patients. His efforts likely led to thousands of lives being saved. He served on numerous public health policy groups, including the NIAID AIDS Research Advisory Committee from 1991 to 1995, NIAID's National Advisory Allergy and Infectious Disease Council, 1995 to 98, and the Fair Pricing Coalition Board of the Foundation for AIDS. Delaney founded Project Inform in 1985 and served as director until 2008, which is based in San Francisco. This organization continues to raise public awareness and lobby policymakers on HIV and AIDS issues. So our participating institutions are the backbone of defeatHIV, the Delaney Cell and Genome Engineering Initiative, fully supporting our collaboratories, research endeavors, and facilitating our scientific progress. Each of our institutions provide unique and complementary strengths that are leveraged in support of the program's activities. Today, we will be focusing on just one of our collaborators, Sangamo Biosciences, which is located in Richmond, California. Sangamo Biosciences is a clinical stage pharmaceutical company focused on the research, development, and commercialization of engineered DNA-binding proteins as the basis of a novel therapeutic platform to address unmet medical needs. The company is the worldwide leader in the development of proprietary technology enabling specific gene modification and regulation of gene expression. Sangamo is developing ZFP therapeutics, zinc fingers, drugs designed to function at the DNA level for treatment and indications including HIV. defeatHIV strategy is to attempt to cure HIV. defeatHIV strategy's attempt to cure HIV has two parts. Number one, the generation of autologous and stable HIV-resistant immune system. We want to make your own immune, which means making your own immune cells resistant to HIV by knocking out the gene that creates the CCR5 receptor and then give them back to you. Number two, direct targeting of latent viral genomes for disruption and inactivation. Essentially, we would like to snip the HIV out of your DNA with special proteins that cut like molecular scissors. So to achieve these two goals, defeatHIV is organized into two major subgroups. Number one, gene modification and the delivery subgroup, then also the transplant and clinical subgroup. These two subgroups are populated by work done by 5 projects and 5 cores, and 1 CAB. Project number 2, this looks at the therapeutic grade zinc fingers nuclease. Targeting the macaque CCR5 is led by Dr. Phil Gregory of Sangamo. And so with no further ado, I would like to introduce to you Dr. Philip D. Gregory. He is the Chief Scientific Officer at Sangamo Biosciences. He has served as the Chief Scientific Officer since July 2009 and Senior Vice President of Research since January 2014. He joined Sangamo in December 2000 as a scientist. He became a team leader in 2001, a Senior Director of Research in 2003, a Vice President of Research in 2005. Prior to joining Sangamo, he was at the University of Munich, Germany. He studied the role of chromatin structure in gene regulation and published extensively in this field. He served as a member of the Scientific Advisory Board of Keynote Symposia since December 2009. He was named by Thomson Reuters as one of the world's most influential scientific minds of 2014, and he earned a doctorate in philosophy and biochemistry from the University of Oxford, and a BSc in microbiology from the University of Sheffield. Now, with no further ado, we'll hand it over to Dr. Phil.
So thank you very much for the introduction, and I hope everybody can hear me okay. So before I start, what I'd like to do is first just make a couple of comments. So the first really is just to thank the collaborators, the funding agencies, but perhaps most importantly, the HIV community and the subjects who've agreed to participate in Sangamo's trials. Without their interest and without their bravery, the work that I'm going to describe to you today would not have been possible. And so I just wanted to start out by acknowledging everybody that's been necessary to achieve what's happened to date. So today, what I'd like to do is to provide an overview of the work that we have been doing using zinc finger nucleases, and I'll describe to you what those are in a second, as a genome editing tool in the development of a potential treatment for HIV. So I think it's very important to start by, let's make sure. So Tom, I don't seem to be able to advance the slide.
If you click on the slide, then you should be able to use the arrow keys.
Okay, got it, yeah. Solved my problem. Let me go. Okay, so to start out then, the HIV life cycle has been extremely well studied, and I wanted to show you this just to highlight a couple of important features. Number one is that we do understand a great deal about the basic virology of how HIV interacts with a cell and infects that cell, including, I was going to point to two things. One is that most of the drugs that we currently have operate within inside the cell in various processes post the process of viral entry. So this process of reverse transcription, the integrase that's necessary for insertion of the viral genome into DNA, and as well as the process of creating new viral genomes, which ultimately create the mature virion. What I'm going to talk to you today about is actually the first step of viral entry, which is the engagement of HIV with its cellular target using receptors. The primary receptor is CD4, and that's shown in the top left of the slide. But the secondary receptor that's extremely important, the so-called co-receptor, is CCR5, and this receptor is necessary for HIV to productively interact with its target cell and gain entry to the cell. A second thing I want to point out on this slide is that as part of the natural biology of HIV infection, when HIV does infect the cell, it inserts itself into the genome, and so this integrated copy of DNA that I'm highlighting in the middle of this blue cell here, this is how the cell establishes something called the reservoir. In other words, a collection of cells and tissues in which HIV is integrated and can become a source of new viral replication and new virions. And so that's going to become important later on. But initially then, just to sort of recap on this slide, I'm interested in making sure we understand that HIV interacts with the cell using these co-receptors and that CCR5 is necessary for HIV entry. So what I thought I'd do, just to make sure that everybody's on the same page, is really to sort of scroll back to 30,000 feet and provide a few terms that we use in gene therapy and in genome editing, just to ground everybody. So if you take a cell out of an individual and sort of examine that cell molecularly, the cell houses a DNA, and in fact, all the DNA necessary to encode all of the proteins that are necessary to create a cell. And that collection of DNA, as shown as chromosomes here, is collectively described as the genome. This is the entire collection of genes encoding all of the proteins necessary to create every different cell in the body. If you sort of drill down, or if you imagine expanding down into the structure of just one of these chromosomes and unpackage that, you get to the DNA itself, which is shown here, and inside the DNA, encoded by that DNA, are the individual instructions necessary to encode a particular protein. And so each one of those instructions, each one of those chapters of the book, if you will, is called a gene. And if that gene is transcribed, so this process of transcription, we can take the information encoded in the DNA and turn it via this process of transcription to mRNA and translation into the proteins that are actually encoded by this piece of DNA, this gene. So when we think about gene therapy, the concept is that by working at the DNA level, we can introduce, or as we'll describe today, subtract genetic information from the cell. And really what we're doing is essentially impacting the proteins that will really be engaged in this case, in HIV, but we're doing so at the DNA level to either add or subtract genetic information to provide the cell with some new capability that we think will assist that cell in resisting HIV infection. And that's really what's shown on the next slide, and I stole this slide from a presentation that Paula Cannon gave recently, which I think very clearly describes what's happening. So when we think about using genes to make a cell that's HIV resistant, you can think about adding a piece of genetic information to that cell that will help stop HIV from replicating, and an example of that might be work that's happening within the defeatHIV collaboratory around the C46 entry inhibitor. And this I'll describe as sort of classical gene therapy, or the addition of genetic information to the cell. But one can also think about removing a gene from a cell that HIV requires, that's necessary for HIV to infect that cell. And in this case, the focus of the presentation will be the CCR5 co-receptor for HIV. And in this mode, the subtraction mode, we're going to talk about the concept of genomic editing. So in other words, literally reaching into the genome and snipping out or cutting out the gene that encodes this particular receptor, CCR5. And that, again, is shown in this slide here. So again, just from a terminology perspective, the goal here is to develop something called a functional cure for HIV. And I think it's worth taking a second just to define, what we mean by a functional cure, and that is that it's the control of HIV replication in the absence of antiretroviral therapy, in the absence of drugs, via arming the immune system. In other words, providing an HIV-resistant compartment that will allow the patient's own immune system to control HIV replication. What it does not mean is elimination or complete eradication of HIV. So the patient may remain with detectable HIV, but it's under sufficient control that the signs and symptoms of the infection are abated. And that's in contrast to a sterilizing cure, in which one might imagine the complete eradication of the virus from every cell and all tissues of the body, which of course requires the total elimination of the viral reservoir, which I've described as being the collection of cells and tissues that harbor the virus, HIV, integrated into their genome. Obviously, this is a much more challenging task, simply because of the number of potential cells and the sites of the reservoir existing in the patient. And so our goal is to develop a functional cure for HIV. And the way I think about this is that the goal really is to turn HIV into any other type of viral infection that the body successfully controls, such as a CMV infection or an HSV infection, in which the virus, in both of those cases, may never disappear from the patient, but the process of the infection is under immune surveillance and is under control, and the patient essentially is unaware of their sort of positive status, if you will. The immune system is completely controlling that infection. So that's our goal. So a little bit on the rationale for using genome editing to achieve that goal. So first, in terms of the natural pathology of the disease, HIV itself targets the CD4 T-cells. These are a particular class of immune cell, and the loss of these cells leads to full-blown AIDS. So the CD4 T-cells are a key cell in the immune response necessary to control infection, not just of HIV, but in fact of any infection. And CD4 counts correlate very closely with disease progression. So falling CD4 counts ultimately are an indicator of the immune system's loss of, if you will, of progress against the virus, and ultimately they will fall to a level that will allow full-blown AIDS. So the idea is that we use genome editing to make a subset of CD4 T-cells resistant to HIV. And so we don't plan to make the entire CD4 population in the patient resistant, but we plan to make a subset of these cells that are resistant. And the concept is that the protected CD4 cells, so those that actually no longer have the CCR5 co-receptor, and therefore HIV can't enter those cells obviously can't support new HIV replication. So the number of targets that HIV has to infect and produce more of itself reduced. The more resistant CD4 T cells that we have, therefore, the lower the HIV levels would be. So obviously if HIV lacks targets to infect, then it can't infect those cells and can't give rise to new HIV levels that can go infect new cells. The protected CD4 T cells can also support or even reconstitute the immune system. So because HIV is no longer removing these cells are permanent or persistent and can allow the immune system to recover to support immunological responses to infection. And in that process, we have the potential to support an anti-HIV immune response. So in other words, that these protected CD4 T cells can not only in general help the immune system, but more specifically could support the immune system's ability to recognize and ultimately eliminate cells that are infected with HIV. And the proof of concept as to how this might play out is provided by Timothy Brown and his situation as an HIV-positive individual who developed an AML, a cancer, that was treated with a bone marrow transplant with cells from a donor who had a natural mutation in CCR5, so-called CCR5 delta 32 mutation, in both copies of the CCR5 gene. And so the cells that he received had no CCR5 on their cell surface, and he's remained aviremic now without the requirement for being on antiretroviral medication for over 5 years. And so this suggests a path for eliminating CCR5 as a target in appropriate cells to achieve a control of HIV. So just a little bit on what the CCR5 delta 32 looks like. We know from a number of studies that the CCR5 delta 32 mutation produces a truncated form of CCR5, which is just shown here. And in fact, this truncated form never properly makes it to the cell surface, and so in fact, the cells are essentially denuded of the CCR5 co-receptor, and individuals who are homozygous for this allele are highly resistant to HIV-1 infection. So, for example, subjects who received blood transfusions from HIV-positive donors remained uninfected if they happened to be lucky enough to have this CCR5 delta 32 homozygous status. In the Timothy Brown case, what happened is that the donor that was used to provide the cells was such a CCR5 delta 32 homozygous donor. And so when those cells were repopulated, when they're transplanted into Timothy and repopulated in the bone marrow and gave rise to the rest of the immune system, the CD4 T cells, amongst other cells, were obviously also CCR5 delta 32 homozygous and therefore were uninfectable. So essentially transplanting HIV immunity into his immune system. So when we think about deploying genome editing as an approach to HIV, I think it's important to consider which cells are the targets. So which cells do we want to actually edit? So what I've just described to you in the Timothy Brown case was hematopoietic stem cells, or so-called bone marrow-derived stem cells, which have the ability to differentiate into all of the different, very specialized cell types of the blood, including macrophages, lymphocytes, but perhaps more importantly, also into T lymphocytes, including CD4 T cells. So it would be possible to think about the hematopoietic stem cell and these bone marrow-derived stem cells as a target for genome editing, and we'll touch on that at the end of the presentation. But really, the purpose of doing that would be to create T cells that are CCR5 modified, and so we can also think about T cells as the direct target. And so that's exactly where we started, was thinking about the T cell as the target and modifying the patient's own CD4 T cells using this genome editing approach to eliminate CCR5 and then return those modified cells to the patient to establish an HIV-resistant component or compartment of the immune system. And so this is what's described on this slide as making HIV-resistant cells by removing CCR5. And so the idea is that we can, a patient or a subject would be enrolled in the trial. We would remove cells, either T cells, which can be removed using a process called an apheresis, which is much like a blood draw, or hematopoietic stem cells, in which case it might be first necessary to use drugs to mobilize these bone marrow-derived stem cells into the peripheral blood first so that we can extract those easily. And then in the laboratory, we can expose those cells to the zinc finger nuclease, and I'll describe in more detail what that is in a second, to destroy the CCR5 gene in those cells. And then we can return those cells back to the patient, and again, it may be necessary, depending on the setting, to use drugs to help condition the patient and allow those cells, either the T cells or the bone marrow-derived stem cells, to properly and successfully engraft. And so this is the process that we've been working with, both with the T cell studies, which I'll focus on, as well as some of the HSC studies, which I'll get to at the end of the presentation. So I'm now going to get into a little bit more of the details on the SP728T work. So SP728 is the designator we give to the nucleases. These are the reagents that are specific to the CCR5 gene. And T here relates to the fact that we're targeting the CD4 T cell population. So the product starts out with nucleases, of course, which target the CCR5 gene. And these reagents are designed to introduce a cut in the genome of the cell at the CCR5 gene. And that cut, when it's resolved via this error-prone DNA repair pathway, results in the introduction of insertions and mutations at the site of the break. And those insertions and mutations can give rise to the disruption of the CCR5 target. And so if successfully applied, these nucleases then drive towards a genome that has a permanent genetic disruption of the CCR5 gene. And so cells and the progeny of the cells derived from these will no longer express the co-receptor CCR5, which HIV uses to enter the cell. There are other ways or many ways at the moment of doing this type of genome editing, we're using zinc finger nucleases. They're effectively designer restriction enzymes. That's a graphical depiction of what the nucleases look like when they're targeting DNA. We actually have to make two different ZFPs that appropriately dimerize on DNA to generate this cutting event. And then we deliver them, in most of the trials I'm going to talk about, we've delivered them to the T-cells in a process that makes use of
Identify who asked the question? Yeah. Dr. Gregory, if you don't mind the brief interruption, we do have a question
Sure.
from one of the listeners, Alistair Burton. And the question is, what is NHEJ?
Good question. Let me go back. So this is it's called non-homologous end joining, and it's the process by which the ends of the DNA shown on this slide are essentially glued back together again by the cell's DNA repair machinery. And the reason it's called non-homologous is it doesn't require any homology in the ends of the DNA, or it doesn't require any template to repair this. It really is simply the gluing back together again of these two ends. And because there's no homology component to this, information, so DNA, can be gained or lost at this site, and the system is blind to that. And so that's why it's error-prone. It doesn't know if there's been any damage that removed information at this site, and so it tends to drive towards disruption. So NHEJ stands for non-homologous end joining.
Thank you so much.
Okay. So just to place the CCR5 ZFN cut site in relation to where the delta 32 mutation is, this is the point of where the 32 mutation is. Here is on the protein sequence where the CCR5 modification is. And so if anything, we would make a much more truncated version of the CCR5, and one in which we've actually not been able to detect its presence at the cell surface. So we think that by cleaving at this point, we make a very truncated protein that is not successfully presented to the cell surface of T cells, and so essentially should be it should function as a complete CCR5 null. To introduce those reagents clinically, we modify the T cells in an ex vivo process, and so we make use of essentially a cold virus called adenovirus, a particular type, the 535, which is successful at infecting T cells. And then more recently, as I'll get to at the end, we've moved over to messenger RNA electroporation. So this is a process that doesn't require the use of a viral vector, and is simpler and potentially allows us to do repeat dosing. And I'll describe why that might be important a little later in the presentation. So the process then is just shown in toto. Yes, absolutely. Dr.
Gray. I'm sorry for the interruption. We just have one more question from one of our listeners as well, and there's actually a few questions here. I'll just begin. Do we have, from Lori Silla from the defeatHIV CAB, do we have any idea what % of cells need to be genetically modified to eventually achieve protection? That's the first question. There's two others. I'll start with that one.
Yes, that's a great question, and actually is one that we don't know the answer to, and we're hoping that the studies that we're doing will shed light on that. I think it's clear that we don't need to have 100% of the cells be CCR5 modified, but it may be necessary to have, importantly, a subfraction of the cells which are also HIV reactive be CCR5 negative, and that number may be very important. There'll be a slide a little bit later in the presentation that'll show a correlation between the number of CCR5 modified cells that we've infused into the subjects and the impact on viral load that suggests that there may be a threshold for successfully controlling HIV. But I'll wait till that slide.
Okay, and the other question was, how long does it take to reconstitute the new immune system?
Well, with the T cell responses, the effects are very quick. So when we infuse the T-cells, they actually expand quite substantially in the patient. And so we see immediate effects on the number of CD4 cells, and we see immediate effects in terms of the number of CCR5 modified cells. And so we think that the impact on the immune system is quite rapid, at least in the context of the CD4 T-cell product. For the hematopoietic stem cells, it does take longer because the infused cells have to be given time to contribute to the differentiation and maturation of the cells before they actually become CD4 T cells. So if you infuse the cells that are at the stem cell level, they first have to decide to engraft, which is important, but they also have to decide to differentiate. And so that might take closer to maybe 6 months to see substantial numbers of T cells that are derived from the infused HSC product.
Much. The other question, do we know what percent of Timothy Brown's HIV-resistant cells were resistant from transplant versus those which were grown out later? And in their last question, are there any implications about macrophage infection or protection if we are focusing exclusively on T cells?
Oh, those are two great questions. So obviously Timothy Brown received a bone marrow transplant from a donor who was a 100% a CCR5 delta 32 heterozygote, and homozygote, I beg your pardon. And so 100% of the infused cells had the property of being resistant to HIV. That said, when we do these transplants, there is always a competition between the cells that arrive in the patient and the patient's own cells. And so initially, there was a period of chimerism, so-called, a mixture of Timothy's original immune system, if you will, and the new immune system provided by the CCR5 delta 32 homozygous donor. But quite quickly, over the course of a few months, those cells, the new cells, became almost 100% of his immune system. And so and that's because that was a so-called allogeneic transplant. It's a foreign transplant of somebody else's cells, and there's a competition between those cells as they arrive.
Okay.
And the second question was?
Yes. The second question they had is, are there any implications about macrophage infection protection if we are focusing exclusively on T cells?
Right. That's a great question. So obviously the macrophage is another a target for HIV infection, and as a consequence, it's another source of HIV replication. And so I think if one considers the goal limiting the amount of HIV that's being generated by these target cells, then protecting the macrophages may well be an important component to the ultimate control of HIV. The counter-argument to that is that the longer-term, functional cells that we need to protect to establish a new immune system that can effectively battle HIV is not so much the macrophage, but really is the T cell. And so we feel that the CD4 T cell approach should be able to provide that specific arm of the immune system that's lacking in subjects that have been exposed to HIV. And if we can restore that arm, then other aspects, including the fact that there are macrophages infected, should be placed under the same control. But that's a hypothesis, and is one of the reasons why we're pursuing both the CD4 T cell approach, which obviously won't directly correct and protect the macrophages, as well as pursuing the stem cell approach, which will protect not only the T cells, but also macrophages, as well as dendritic cells, which may be ultimately important in establishing a full immunological control of the virus.
Thank you.
Okay, so the slide in front of us right now is the product. I just want to walk you through this a little bit. subject on the right here, we perform an apheresis. This is it's performed at a sort of blood donor type setup. It takes a few hours to provide the T-cells, which are then sent to a centralized manufacturing facility in which we first purify the CD4 T-cells. We then expose those cells to the CCR5 nucleases. This is all done ex vivo in the laboratory. We confirm that we have sufficient modification, and we expand those cells. So T-cells grow ex vivo very prolifically, and so we can grow these cells to the point where we have sufficient cells to infuse between 10 and 30 billion CD4 T-cells back into the patient. So the way this is done is that we actually grow these cells up to the point we have this number of cells, and we freeze them, and then we do a series of tests to ensure that the cells are exactly what we think they are, and are pure and sterile, etc. And once the product has passed all the release tests, we schedule the patient in for this infusion process, and the infusion actually takes is quite quick, just a few 30 minutes to an hour or so, and then we'll keep the patient in the doctor's office for another couple of hours just to make sure everything's gone well before releasing them home. So it's a relatively simple process for this T-cell product. So in the development of the SB728T, we've now performed several key exploratory clinical studies that have attempted to make use of the information that's become available for each of the as we've continued the clinical development of this product, and attempted to refine how we are both deploying these T-cells as well as refining the patient populations, and how we're exploring the potential of these cells to impact HIV. So what I thought I'd do is take you through different questions that we've, on the previous slide is sort of the different names of the trials, the different studies that we've done, but really as you develop a drug for HIV, these are sort of the important questions that you try and address. So first and foremost, is the drug safe, and is it tolerable to the patient? The second, given that we're yes, please go ahead.
I'm sorry, Dr. Gregory. We have just another question from one of the listeners, from Alistair Burton. What volume approximately is 10 to 30 billion?
Yeah, so that's it's maybe half a liter, something like that. It's not very much. It's okay. So they're really quite concentrated when they go back into the patient. And just to put in perspective, how many, it might be important to understand how many T cells you have. You have, on average, a healthy individual might have over 100 billion CD4 T cells in total. And so this is, it's a substantial fraction, but obviously just 10 to 30% of the total CD4 cells that the patient would have.
Okay, we have another question here from Morgan. What are the immune effects of the apheresis where the patient is missing a significant number of immune cells? And would this pose a greater immune risk to the patient as they wait to receive the modified cells?
That's a great question. So, and it relates to the answer I've just given. Our goal in doing the apheresis is to extract in the range of 100 million or so CD4 cells out of the patient's total of 100 billion. So it's a very small percentage that are taken out, relative to the total number, and then we can put so many back in because we can expand those cells ex vivo. So the actual risk in terms of the number of T cells that are removed is relatively low. This is not a significant fraction of the total T cell complement that the patient has prior to the apheresis.
Thank you.
No problem. Okay, so the key questions that we're trying to address, as I mentioned, safety and tolerability. We're infusing CD4 T-cells, so we obviously are interested to see whether there's an enhancement in CD4 T-cell numbers. We're interested in understanding whether the modified cells engraft. That means that when we infuse them, that they persist and stick around. It's important that they're functional, and we can test that in a couple of different ways. We can ask whether they traffic to important reservoirs of HIV infections, such as the gut tissue, and we can ask whether they contribute to normal functional T-cell responses. So it's important that these cells are not only modified, but that they act and behave like regular CD4 T-cells that will do their normal job, namely control infections of every type, and hopefully also HIV infection. And that's the last point, that we want to understand whether these infused cells have an impact on the viral load. So in terms of the of SB728 and key exploratory studies, I'm going to initially start with this 902 study that was performed in immune non-responders. So these are patients who have low CD4 counts despite being on antiretroviral therapy. And we've studied these patients now for over 3 years. And I think that importantly, so this allows us to ask questions about the CD4 T-cell reconstitution after we've infused this SB728T product. So each of these lines represents a different patient. What's being shown here on the left is the change of CD4 T-cell count. So not the so 0 means no change. 500 means 500 more CD4 T-cells per microliter of blood. And so you can see in all patients early on after infusion, we see very robust increases in CD4 T-cell levels, and that those are maintained even 12 months after the infusion. So we see very good, very robust increases in CD4 T-cells, and this is just quantified on the right-hand side here. And so the mean increase shown at the bottom is 103 cells per microliter at 12 months, and that's important because you'll notice that baseline, some of these patients had, on average about a CD4 count of 400. So that's, a 100 cell increase over a starting level of 400, or in other words, about a 20% increase.
Gregory, question from Dr. Gregory. A quick question from Rick Walton. What do you consider a low CD4 count?
Yeah. So great question. So it depends on for this trial, we placed a line at a CD4 count of 500, and we enrolled subjects that were above 500 into one of the cohorts, and between 250 and 500 into the cohort I'm describing here. And so low for us was less than 500, and that was simply how we defined sort of an immunological non-responder. The normal CD4 counts for a sort of healthy individual are around about 1,000, and so even you could argue that 500 to 750 is still lower than normal, but for the purposes of how we enrolled subjects and how we moved them into these different studies, we took a less than 500 as being sort of a low-ish CD4 count to enroll in this trial.
Thank you.
So we are also interested, of course, in monitoring what happens to the CCR5 modified cells, and to do that, we made use of the fact that there's a specific deletion or a specific modification that we can measure in the DNA of the cells that are modified. And so this gets very technical for a second, but I'll just describe this. These underlined regions are the regions to which the ZFPs actually bind. And so the bit in the middle is where the cut site happens. And these black bases here are types of insertion and deletion that we see. And it turns out that there's a very common repair event when the nuclease is cut that causes a duplication of the 5 base pairs in between the 2 nuclease target sites. And that duplication, just for in internal slang, became known as the pentamer duplication because it has 5 base pairs. And that sequence we can measure directly, so it allows us to have a molecular signature of cells that have undergone our process and that have CCR5 disrupted. And so in the slides that follow, I'm going to describe this sort of pentamer modification, and that's what it is. But really, the way to think about it is we're just measuring the numbers of ZFN-modified cells. And so it gives us a way of assaying how the modified cells are behaving in these subjects. And so if I show you the next slide, here we see the long-term detection of CCR5 modified peripheral blood mononuclear cells. So these are white blood cells from essentially a blood draw post-infusion. And you can see that we see so from infusion we see a nice expansion of these cells. And again, this actually persists over this is now 24 months and even out to 44 months in the patient that's gone out the longest. And so these cells are actually quite long-lived, even though they're modified at the CCR5 locus. And in these subjects, the patients never came off their antiretroviral medication, so the HIV should be suppressed in these patients the whole time. But the persistence of these cells is a little surprising. If you think about T cells as being a cell that that turns over, that doesn't last very long, and so it suggests that we're actually modifying cells with a capacity for self-renewal, and that actually could have very useful properties from the perspective of the durability of the responses we'd like to see. And actually, we went back to look at the products, so the SP728 T cells, before we infuse them into the patient, and found that in these SP728 T cells, this is the CD4 population here, you can look at the different types of cells that are present in this product, and importantly, compared to the pre-modification cells, the post-modification cells see a big expansion in this red sector, and that red sector are the cells that have a so-called memory stem cell-like phenotype. So let me go back and just describe what this means for a second. T cells have multiple different types of T cells. There are effector cells that are actually sort of committed cells that are going to go off and try and do something about an infection. But once you've been exposed to an infection, your immune system likes to remember that. And so these memory cells are one way in which your immune system remembers what it's been exposed to. And so their function is to persist, and if you're ever re-exposed to that same infection, then those cells will be present, will remember quickly, and be able to sort of rapidly advance against this repeat infection. And so these memory stem cells are really quite important because they're both a source of T cells, and therefore, if we've modified them, they're a source of CCR5 modified cells. But perhaps also importantly, they're very long-lived, and so they're able to support the production of other T cells for long periods of time. And if we look to see in this T cell stem cell memory population, so this is this very long-lived population, and we measure the CCR5 modified cells, we see actually that these are the cells that are expanding most. So the median fold expansion of the CCR5 modified cells relative to how much was infused is about 20. So we have 20 times as many CCR5 modified cells that are in this compartment, this long-lived stem cell-like compartment. And in contrast, the effector memory, for example, is only enriched by about threefold. So that's a very significant enrichment in this potentially very important T cell population. And that duration of is shown is quite persistent. So now I'm showing you data. This red line, this is the T cell stem cell memory, either committed or naive cells. And you can see from month 9 all the way out to, in some cases, month 44, we see a very persistent and stable level of modification this is now CCR5 modification in those cells. So we're seeing very stable levels of modification in this critical T-cell stem cell memory population. I also mentioned that it was important to ask how these cells behave, and do they behave like normal T cells. And one of the areas that's very important in HIV is the rectal mucosa. This is a known key battleground of HIV infection. There's a lot of T cells in the gut mucosa. And so it's important that the modified cells that we have should also behave like normal T cells and should traffic to sites like the gut mucosa, just like normal T cells, and that's exactly what we see. That at baseline, obviously, we don't find any of these cells, but after infusion, up to 48 weeks, which is the longest we've looked, we see the presence of these cells in gut biopsies and in the rectal mucosal tissue in subjects that agreed to allow those samples to be taken. And so this shows that these cells are able to traffic to these important regions of the for HIV infection. And then, actually, as a consequence of those studies, there was one sort of anecdotal patient who was studied, and that also gave us more clues that the cells that we were infusing were functional. So, in this particular patient, I'm now looking at just one patient, and these are these markers here relate to activated CD4 cells. So these are measures of the amount of activation of the CD4s, and the more activation they have, the more likely you are to have an inflammatory response in reaction to some type of infection. And so we don't think this was an HIV driven inflammation, but it shows you that over the course of these measurements, we see an increase in the number of cells that are positive for this highly active state, and that was true both in the CD4 population as well as the CD8 population. And so, given that we saw this increase in activated cells at the 3 and 6 month point, we could also ask whether the CCR5 modified cells were also involved in this inflammatory response. And that's what's shown here, that we see that in the CD4 population, this is now the percentage of CCR5 disruption in these mucosal CD4 cells, that we see this big increase in cells that are modified at CCR5, which of course disappear as the inflammation resolved. And so that is the first human evidence that the cells that we've infused, these T cells that are modified at CCR5, can traffic to the gut mucosa in response to a presumed infection, participate in an inflammatory response, and then resolve as that inflammatory event disappears. And so that's an extremely important observation, as it really is the first evidence that these cells that we've infused are behaving just like normal T cells and can contribute to the immune response to some type of infectious event. So what I've shown you so far is that across all the patients we've treated, the infusion has been well tolerated. In fact, the major things that the patients note are as a garlic body odor associated with a preservative that we use to keep the cells in their frozen state and healthy and happy. But other than that, the cells have been very well tolerated. We do see CD4 T cell enhancement, so we see the numbers of CD4 T cells go up, and we see them stay up for quite a long period of time. We can see the modified cells in graphs, so we can measure them both at early points, and we can see them at late points after infusion, so they last a long time, partly as a function of being in this T cell stem cell memory population, which is very important. We can see them traffic to HIV reservoirs, including the gut mucosal tissue. and we can see them contribute to functional T-cell responses, as I just showed. So this is all very positive. It's what you need to see. Of course, the real question is, what about the impact on HIV viral load itself? And so I'm going to go through the first evidence of an impact on HIV. It was performed at the University of Pennsylvania in one of our first studies, this phase 1 study, in which a small number of patients agreed to participate in a treatment interruption. This means that they stopped taking their antiretroviral medication for a prescribed period of time, in this case about 12 weeks. And so this data has recently been published, and so I'm not going to go through it in huge detail, but this was the first publication in the New England Journal of Medicine describing a genome-edited therapy. This is ZFN-modified CD4 T-cells infused in man. As I've just mentioned, the infusions were shown to be generally safe and well tolerated. Again, marked increases in total CD4 T-cells were noted. We also see traffic to the gut in this study. We also show that the modified cells have a selective survival advantage during antiretroviral drug interruptions. So as HIV returns and starts infecting and killing the unmodified cells, we can see a selective survival advantage, and I'll show you that data. And importantly, one subject controlled viral load to below levels of detection before we began reinstating the antiretroviral drug regimen. And this patient obviously was our star patient from a therapy perspective, because it also generated a large number of ideas as to how to do this, hopefully for more patients more of the time. So I'll just show you that this is a complicated, but I'll share it with you. What we're looking at here is viral load in the peripheral blood during this over time. The treatment interruption period is shown in this gray box. So it started at about 28 days and ended at day 112. And then the LOD here, this is the limit of detection of sensitivity of our assay for HIV presence. So if a point falls below this line, the assay basically can't tell whether HIV is present in that sample. And so what you see when all of the patients come off their antiretroviral medication is that HIV comes back. And this is because of the reservoir we discussed right at the beginning, that cells that contain an HIV integrated into their genome, now that HIV wakes up and begins to go about its life cycle, so it begins to generate new virions which leave the cell and become detectable in the peripheral blood. And so that happens in all patients. But importantly, in several patients, we were able to see a decrease in viral load from peak before we began antiretroviral medication. And importantly, in one patient, this green patient here, despite being clearly positive at 2 points, by the time we took this last sample and then began their antiretroviral drug medication again, the viral load was down to below the level of detection. So this patient was capable of suppressing a virus by themselves. And so this became very exciting to us because this was what we'd hoped to see. And it turned out in further analysis that this subject had one of the two CCR5 alleles was a CCR5 delta 32 allele. So in other words, this patient was heterozygous, had one allele that was already CCR5 mutated, and so our job was half as difficult. We only had to make the other CCR5 allele modified to make cells that were completely resistant to HIV. And so this generated the hypothesis that perhaps we were just below the levels of CCR5 modified cells are really important to achieving this low level of this control of HIV. And so if you look at all the patients and ask what happened to the CCR5 modified cells relative to the unmodified cells during this treatment interruption, we see exactly what we'd hoped for. And that is that during the treatment interruption, as I showed you on the previous slide, HIV returns and starts infecting T cells, and so we see a reduction in T cell count during this period. I should point out that at no point does the T-cell level drop below the starting levels. They're still higher than the starting levels because of the remarkable increase in CD4 T-cells that we see in these subjects. So at no point did the CD4 levels actually go below baseline. In fact, they stayed above baseline. But importantly, for the CCR5 modified cells, there's really a much dampened reduction in those cells over time, despite the fact that during this period there is HIV is present and infecting and killing the unmodified cells. And so this is the first human evidence that these CCR5 modified cells are indeed resistant to HIV infection in man. And so it was an extremely important observation, as it sort of was the first human evidence of what we'd seen in our preclinical studies in mice and in the test tube. So as we've been thinking about how do we I'm going to go back 2 slides here. So how do we think about this patient, and how do we think about how to do this more often? One of the obvious questions is, and this actually came up earlier in the conversation, what levels of biallelically modified CCR5 edited cells do we need to achieve this viral control? And so what this graph shows is the change in viral load shown on this axis, along with the number of CCR5 modified cells on the bottom axis here. And obviously what we can see is that these 2 patients stand out. They have both the biggest change, so this is a greater than 2 log change in viral load, but they also have the highest modified CCR5 levels. And so that suggests that we have to get into this range to achieve this range of CCR5 modified cells to see these types of changes in viral load. And so we've been thinking about how to go about doing that. And so that's sort of described on this slide. If more CCR5 modified cells equals better viral control, then the question is, how do we get more modified CD4 T cells to engraft in our subjects? And so we've been exploring essentially 3 different paths. The first is to recruit CCR5 delta 32 heterozygous subjects. So just like the patient in the PEN trial I've just described that became aviremic in the absence of antiretroviral drug regimens, that individual was a CCR5 delta 32 heterozygous subject. So can we just accrue more of these? And for each of these individuals, we effectively get a 2x increase in the number of CCR5 modified cells. It's a doubling of the CCR5 knockout rate. The second thing, yes.
Dr. Gregory, thank you so much. Thank you so much for the extremely insightful and very informative presentation. We are coming up on our time, and I just wanted to kind of give a notice of we have about 10 more minutes for the presentation, and then we wanted to allow at 10:20 about 10 minutes of questions and answers there. We have a lot of questions coming in as it pertains to your presentation, so we want to give some folks the opportunity to ask those questions. So we have about 10 more minutes, and then we'll open up for Q&A. Will that work with you?
Yeah, absolutely. We can. I think there'll be a natural stop point that I'll aim for, and I'll be happy to take any questions.
Absolutely. Thank you so much.
Yeah, no problem. So just coming back to this slide, so if more CCR5 modified cells are better, then we can accrue these CCR5 delta 32 individuals. We can also use a preconditioning protocol to increase cell engraftment. So this is what we've been doing with a drug called Cytoxan that essentially, put most simply, makes some space for the T cells to be received. And we can also do repeat dosing. So we can infuse more than one product into the patient, and this is enabled by a switch of the manufacturing from the use of viral vectors to the use of messenger RNA. So with the CCR5 delta 32 heterozygous study, this is the so-called 902 cohort 5 study. We've been looking at that, and I beg your pardon. So it's going to show you one result from that study. This is our star patient, patient 502, who has controlled their viral load for, at this point, 59 weeks at a level of less than 500 copies per mil. The subject remains off of antiretroviral therapy, and so we view this subject as a demonstration that these T-cells can provide a durable functional control. And so the data shown here is a little difficult to see on this, but the black dots are the viral load, and you can see that they've remained no more than about 500 copies at this stage. And so this is a subject who has CCR5 delta 32 heterozygous as the criteria for enrollment. We've also been looking at the Cytoxin preconditioning protocol. So the first question here was how much Cytoxin to use, and here we've been taking a leaf from the cancer immunotherapy playbook, where they also use Cytoxin to improve the engraftment of T-cells in anti-cancer approaches. And so our first question was how much Cytoxin to use, and so what you can see here across the bottom are different doses, 0.1, 0.5, 1, 2, and then actually 1.5 grams, so a step down. And what you can see is that both in terms of the CD4 change from baseline, so how much increase in CD4 levels we see, as well as how many modified cells we get. The pentamer duplication, of course, is a measure of how many CCR5 modified cells we put in. The optimal turns out to be about 1 gram. So we see the biggest CD4 changes. So now over a 3,000 increase in CD4s per microliter if we use 1 gram, and about 120 pentamer duplications per microliter again if we use 1 gram, and that's better than all of the other conditions. And then we've also been, in some of those subjects have agreed to participate in a treatment interruption, and in several subjects we see now a 2 log or more decrease in viral load, and we have several subjects that are continuing their TI, so they have maintained their CD4 levels and have HIV levels that have not triggered a return to antiretroviral medication. But this suggests that the Cytoxin-based approach may also have the capacity to provide enough CCR5 modified cells to drive control of HIV. And so these are just 2 of those subjects. Again, the graphs are a little bit difficult to see. The black dots, again, are the viral load, and you can see this reduction from peak here at the beginning of the TI. That's about a 2 log reduction, and this subject, again, here's the peak, and here's the current situation for this patient. Again, about a 2 log reduction in viral load, and in both cases, the subject remains off ART. The subject on the left here, these 04019, has been off ART now for 39 weeks. So it's, again, a very long and sustained control of HIV. As we think about the HIV, what I've been showing you so far is the control of the viral load as measured by viral genomes that are present in the bloodstream. But the other component to consider is the HIV reservoir itself. In other words, the cells that contain an integrated copy of HIV and can be a source of new HIV infection and are essentially a way in which the virus maintains itself. So we've been interested in whether our approach, which should support immunological detection of these cells without reinfection, could also have an impact on the viral reservoir. And so that we've been looking at in this first cohort again, which are immune non-responders. They remained on ART the entire time, and we followed these patients for up to 3 years. To measure the viral load, you have to actually measure proviral DNA, so the integrated viral genome. We use a digital PCR assay to do that. It's a very sensitive assay. And I'll just cut to this chase here that across the 9 subjects enrolled in this trial, you can see the slope is negative in all cases. So we see a reduction in in the number of proviral copies. And in several of these subjects, there's been a log or more reduction in the size of the proviral reservoir in these subjects. So that's very encouraging data over this 3-year period. Just to show you that's not necessarily standard. Here's work that was described at CROI and has recently been published on interleukin-7, in which IL-7 also produces a big increase in CD4s, so you can see a nice increase in CD4s here. However, you get a concomitant increase in the viral reservoir, so just as the CD4s go up on the left, so the viral reservoir expands on the right. So what I've just shown you is CD4s getting much more common, or increasing the CD4s as we can with our T-cell product, but a reduction in viral reservoir has not been seen before. Importantly, in trying to understand the mechanism of this, we've found that there are functional HIV GAG. So GAG is an HIV protein that the CD8 cells can recognize, and we see CD8 T cells. So these are the cells that CD4s help to eliminate infection. We see that in subjects that have the biggest reservoir reduction, we see higher numbers of cells that are expressing these immune responsive cytokines in response to GAG. So this suggests that the reason why the reservoir is reducing is that we have in these subjects been able to support an anti-HIV or immune response. So I'll probably stop on this slide, but just to sort of review what I hope I've shown you. Obviously, we couldn't perform these studies unless we were able to manufacture this product. So we've been able to achieve consistent manufacturing of these CD4 T cells. To date, we've now treated over 70 subjects with CD4 T cells, and across all those studies, the drug has been generally safe and well tolerated. We see very consistent increases in CD4 T cells, which are quite durable over time. The modified cells engraft and persist nicely, partly as a function of the fact that we're, as I mentioned, infecting and protecting these stem cell-like T cells. The cells are functional in that they traffic to HIV reservoirs as well as contribute to functional T cell responses. And we're actually seeing an impact on the viral load and on the HIV reservoir. And our goal really is to try and figure out how to do this how to make this a more robust across the populations that we're treating. And so, perhaps in the interest of time, I'll stop here and open up to questions.
Thank you so much. The first question is from Alistair Burton, and this is excuse me, before I pose this question, just to let folks know, what we're going to do is ask that you submit your questions via writing or email, via text rather. We will answer them in the order in which they've come in. For those questions that we don't have an opportunity to answer, we will get them to Dr. Gregory, get his answers, and we'll post those via email that we place on Twitter. Also, we'll place on Facebook, so to ensure that all of your questions are answered. So we'll go back to the first question. It's from Alistair Burton. What is an estimate for average CD4 lifetime?
An estimate for average CD, what was the question, sorry?
I'm sorry, what is an estimate for an average CD4 lifetime?
Right, so that depends on the type of CD4 cell. If it's a CD4 effector cell, so one that's sort of committed to participating in an active response to an infection, that might have a lifespan on the order of weeks. The central memory population that we described, that we're modifying very successfully here, has a half-life of about 7 years. So it would take about 7 years for you to need to replace those. So depending on the population, it can be days to weeks, or depending on the precise CD4 subtype, it can be days to weeks, all the way through to maybe 7 years.
Next question comes from Alistair Burton once again. Is there any thought of trying for zinc finger treatment to include both CCR5 and CXCR4 deletions?
That's a great question. We have actually begun doing that. So we've published on reagents that we've made that target the CXCR4 co-receptor and can show that those cells are, as one might predict, resistant to X4 tropic virus. And we have gone on to show that it's possible to dual modify cells, CD4 T cells, so that they don't express either CCR5 or CXCR4, and that those cells are, as you might imagine, resistant to both X4 and R5 tropic viruses. In the current studies, the way we deal with that problem is that we conduct pre-screening of subjects to ensure that they have no detectable X4 virus prior to enrollment in our study, because obviously a CCR5 targeted therapy won't protect against an X4 tropic virus. And one of the stopping rules for our studies is that if an X4 virus is detected, and the subject should be returned to the standard antiretroviral therapies.
Thank you so much. Another question from Rick Walton. How did you acquire the FDA approval for treatment interruption?
Great question. So yes, this is a very obviously a very important component of all cure-based strategies. Is that at some point you have to test your hypothesis by removing the current standard of care, which is antiretroviral therapy. The way that we've approached this is to do this in a very stepwise fashion. So we initially in our first studies did a very short treatment interruption of just 12 weeks, and with very strict stopping rules if the CD4 counts fell below a certain level, or if the viral load was above a certain level, then we would reinstate medication. And during that period, as I mentioned during the presentation, we were able to show that because of the big increases in CD4 cells that we obtain following infusion of our product, we never exposed the patient to less CD4 cells than they had to start with. They always maintained actually an improvement in the number of CD4 cells. And that data, combined with the general safety of performing the treatment interruption, was reviewed by the agency, and in subsequent trials, we were able to extend that period of interruption to 16 weeks, and then ultimately to a more permanent treatment interruption, again, so long as the patient's CD4 levels and viral load do not pass certain thresholds. And obviously we measure those very carefully, and our goal is to make sure that the patient is always doing better than they were beforehand, and if not, immediately returned onto medication.
Thank you so much. The next question comes from Michael Allen. Can you elaborate on the theoretical benefit of multi-dosing to increase the percentage of biologically modified CD4 T-cells?
Yeah, so the idea here is that by switching to the mRNA product, we should be able to infuse the T-cells multiple times. So what we see when we infuse the T-cells is a big increase in CD4 T-cell numbers. So the thought is that if we either split the dose or, in fact, use multiple doses, we should be able to, with each dose, infuse more T-cells and get more of our cells engrafted to achieve higher and higher levels of total CD4 cells that have our modification. And so those studies are, in fact, underway. The mRNA study is now open and is accruing patients. So we're already making progress towards answering whether that hypothesis will play out in practice.
Excellent. Thank you so much. I have a question. With regard to HIV cures, and I do say cures plural, what are your thoughts on the scalability and the implementation of cures when we do finally have them, as pertains to the masses? Scaling a broadly accessible cure for HIV. What are your thoughts around the scalability and what that would look like?
Yeah, so obviously the autologous cellular therapy is it's both an expensive and a complex proposition in its current format, and that's both that's a function of it being a sort of a patient by patient process, and it's also a function of us wanting to do the best possible product that we can in the most controlled way that we can for these early clinical trials. I think what you'll see if the data support the advancement of these types of therapies, I think what you'll see is investment in both the process and the process development that will be just like the process and process development that was necessary to begin to think about scaling antibody manufacturing from the initial clinical trials that were done to the industrial scale 5,000-liter manufacturing processes that exist today. When those first antibody therapy studies were done, no one was sure that it would be possible to manufacture antibodies at the scale that would be necessary. And I think that we're, honestly, as a field, we haven't had, to date, the data that has driven that level of investment. But my belief is that as we make progress with these therapies and as we show that these are approaches that can give rise to the types of cure-based outcome that we hope for, that will drive investment in the process development to bring down the costs and then will make this therapy available to more people. Along those lines, an ultimate goal might be to eliminate the need to take the cells out of the individual altogether. So would it be, to try and think about whether it would be possible to perform the editing reaction inside the body, and that might be a much more scalable concept, and that's work that's currently underway in numerous laboratories at the research scale. So we are looking at ways in which, if this is successful, how might we develop this in a way that would make it both cost-effective and scalable, both within the Western world and ultimately, of course, across the Third World.
Dr. Gregory, we cannot thank you enough for your time and this invaluable information. What a wonderful presentation, and it's been great to have you with us today. We want to thank you on behalf of the Defeat HIV CAB for your time, and we look forward to being able to have you present again and working with you once again. So thank you so much for this invaluable information.
Yes, thank you, and it was my pleasure, and I look forward to participating again, and obviously if there's any questions I can address offline, I'd be happy to do that.
Absolutely, and we'll definitely get those questions to you. Also, on behalf of the Defeat HIV, we would also like to thank Tom Andrus, one of our Defeat HIV CAB members, who makes much possible for us, including this webinar here today. Thank members of the DARE and CARE CABs, and they are part of the larger NDC CAB collaboratory. And all of these participants, you who are out there, have been listening to us and asking wonderfully insightful questions. We thank you so much, because obviously without you, this webinar would be impossible as well. So thank you so much, folks. Have a wonderful afternoon.